Here we present deep learning-based approach, host depletion of metagenomic samples. Metagenomics is a study area where we collect samples from different environmental bodies and explore the microbial diversity and their functionality in those samples. One of the main challenges that we face in this analysis is the presence of host DNA that overshadows the microbial reads. So, it is really important to remove these host DNA sequences from the sample. There are different methods already available for this task, but these methods either use laboratory techniques or alignment-based methods where they align the DNA in the sample to reference genomes. Both these methods have low accuracy and are costly and time-consuming. Therefore, we developed a machine learning-based tool that provides a more accurate and resource-efficient solution for host depletion. This tool is based on a feed-forward neural network model, and it consists of 5 fully connected layers. It takes a feature vector as input and outputs a classification label that indicates whether the sequences belong to the host species or to a microbial species. DNA sequences were encoded into kmer frequency vectors, where we used combination of 2-mer and 3-mer frequency representations. We trained and validated our model using 2 datasets of 250,000 reads each. Half of the training data comprised simulated reads randomly sampled from reference genomes, 50% human and 50% from 5 microbial classes, with species counts and read distributions shown in the table. The other half consisted of nanopore reads, equally split between human and microbial classes. Reference genomes were sourced from NCBI GenBank and nanopore reads from NCBI SRA. After tuning model parameters, we tested the model on metagenomic datasets from PacBio and nanopore platforms. We compared performance against Kraken-2, a leading taxonomic classifier, and AMAZE, a CNN-based host depletion tool. Our model showed the highest accuracy and specificity on simulated PacBio reads. High specificity is critical to avoid misclassifying microbial reads as host, which would hinder accurate microbial composition analysis after host depletion. Then, we tested our model on nanopore datasets with varying host percentages of 1% to 99%. Our model had the highest overall accuracy when the host fraction was below 50%. However, after that, Kraken-2 and AMAZE's accuracy significantly increased when the host fraction increased. However, here also, our model had higher specificity through all host fractions, indicating its ability to deplete host sequences while preserving microbial sequences, ensuring accurate microbial analyses. In terms of resource utilization, our model significantly outperforms these 2 models. For classification, Kraken takes around 87 gigabytes of RAM usage, whereas AMAZE used around 5 gigabytes of RAM, and our model needed only about 3 gigabytes for these test datasets of size 24 gigabytes. However, in terms of classification time, AMAZE took around 4 hours, while our model took only about 10 minutes. Therefore, in terms of resource utilization, our model is better compared to those tools. We also compared our model using a real metagenomic dataset that is publicly available in the NCBI SRA database. For this dataset, our model demonstrated an accuracy of 87%, while both Kraken-2 and AMAZE had an accuracy of 94%. Here also, we had 99% specificity. To provide a fair comparison of resource utilization, we trained the AMAZE model on the same training dataset. The training process for AMAZE took approximately 44 hours, while the proposed FFNN model was trained within 12 minutes. In terms of peak RAM usage, the FFNN model required only 0.93 gigabytes, whereas AMAZE utilized 5.92 gigabytes. This demonstrates the effectiveness of feed-forward neural networks with kmer encoding for host depletion, offering a more efficient alternative to CNN architectures using one-hot encoding, which are more computationally demanding during both training and classification. Therefore, our approach for host depletion using normalized kmer frequency vectors for sequence encoding provides a more resource-efficient and generalizable alternative to existing host depletion methods, which contribute to performing metagenomic analysis in resource-constrained environments.